So as in the last video we have discussed that how to find the net pressure which is present in the glomerulus. Alright, so in today's video we are going with the second step of urine formation, that is tubular reabsorption. So let me write here the topic, tubular reabsorption. Reabsorption. So this is the second step of urine formation. Alright, and reabsorption will be done by the help of PCT. Alright, so that is proximal convoluted tubule. So let me make here the structure so that it can be more clear to you. So firstly I am going to make here the Bowman's capsule. This is Bowman's capsule. Wait. This is Bowman's capsule and this is PCT. Alright, so I am going to make the cells of Bowman's capsule and PCT. As we know that in the Bowman's capsule we have squamous epithelial, but here when they are reaching there are modified squamous epithelial which are also known as podocytes. Alright, so here are squamous epithelial cells in the Bowman's capsule. These are all squamous epithelial. Alright, so in the PCT we do have cuboidal cells. Alright, cuboidal epithelial cells, and they have a big surface area. Alright, these are like this. These are like this. Alright, and here I am going to make a big one so that I can explain you reabsorption. This is the cell. Brush border cuboidal epithelial. So let me write here 2. This is what brush border cuboidal epithelial. Cuboidal epithelial. Brush border cuboidal epithelial are the cells of PCT. Alright, so this is Bowman's capsule where glomerular filtrate is present. Alright, so let me write here down with white one, not with white but with this color. Alright, so here what is present here? Glomerulus filtrate is present. Filtrate. So let me draw here as well, glomerular filtrate. This is basically glomerulus filtrate. Alright, so now reabsorption will be happened by the PCT, which is proximal convoluted tubule. Alright, and in the PCT we do have cells, cuboidal cells, and these are modified, which are known as brush border cuboidal epithelial. Now we have a membrane here. Wait, let me make here. We have a membrane here which is known as apical membrane. Alright, which is known as apical membrane. So let me write here down. Where should I write? I don't have space. Alright, I can write here, or I can write here. So this is what? Apical membrane. This is apical membrane. Alright, and we do have membrane here as well, which is known as basolateral membrane. Basolateral membrane. So let me write here down. This is basolateral membrane. Alright, so there are two membranes. The first membrane is known as apical membrane, and the second membrane is known as basolateral membrane. Now we do have pump here. Alright, in the brush border cuboidal epithelial, we do have pump here. So I am going to make this pump. Here we have pump, which is known as sodium potassium pump, which is known as sodium potassium pump. So here I am going to write sodium, alright, potassium pump. So now we know that in the glomerulus filtrate there would be sodium ions as well. Sodium ions would be there as well. So let me write here down sodium, wait, sodium ions would be there as well. So sodium ions have to go out. Alright, so here I am going to make the paratubular capillary in which it would be absorbed, reabsorbed. So I am going to make here the paratubular capillaries. Where should I make? Okay, this is fine, please. This is what? Paratubular capillary. Paratubular capillary. Alright, this is paratubular capillary. So now it would be reabsorbed, sodium ions would be reabsorbed in the paratubular capillaries. So now sodium potassium is like a pump where 3 ions would be reabsorbed by the paratubular capillaries and 2 potassium ions will get entered to the PCT. Alright, so it will actively extract sodium ions to the paratubular capillaries and potassium 2 potassium ions to the PCT. So here I am going to sodium ions would be here, 3 sodium ions would go there and 2 potassium ions would go there, would go inside it. Alright, so let me write here down as well. 2 potassium ions and it is throwing 3 sodium ions. Wait, it is creating mess now. So but 3 sodium ions are going to the paratubular capillaries and 2 potassium ions are going to the then PCT or proximal convoluted tubule. Alright, so here I am going to make here now. Alright, in the first cell we don't have place. So now here we have aquaporins, which are used for absorbing water and then sending to the paratubular capillaries. So this is what? This is aquaporins. This is the protein. Aquaporins. Alright. This is aquaporin and this is not visible, I guess. I should remove it and then write. Alright. So this is aquaporin. So this is aquaporin from where water will be going inside. All right, water will be reabsorbed by the help of aquaporin. So I'm going to write this with white color. So water would be absorbed here as well. So now what is remaining? Glucose. So here we have one co-transporter which will take sodium as well as glucose to the paratubular capillaries. So here what we have co-transporter. Co means with that, and transporter means to transport. So these both are transporting together glucose, and we do have uh, sodium ions. So I am going to make this with, which color should I use? Alright, this color is fine. So here we have co-transporter from where sodium ions plus glucose would be going. Sodium plus glucose would be going to the paratubular capillaries and it would be absorbed. And these are brush border cuboidal epithelial I have already written. So now the question is, how much water is absorbed from the PCT to the paratubular capillaries? So 99% of the reabsorption is done by the PCT. Like 70 to 80% of water plus ions are reabsorbed by the PCT. So maximal absorption or reabsorption is done by the PCT. Then we would have reabsorption in the DCT, distal convoluted tubule, as well, but maximum reabsorption is done by the PCT or proximal convoluted tubule. So I am writing here with this color. All right, maximum reabsorption absorption is done by PCT or proximal convoluted tubule. By PCT or proximal convoluted tubule. And what's the percentage we do have? 70 to 80% water plus ions. So let me write this with this color. 70 to 80% water, H2O, plus ions, sodium potassium ions. All right, ions, water and ions are reabsorbed by the PCT or proximal convoluted tubule. All right, so in the last video, as I told you that we'll be discussing about that why urine is yellow. So now we are going with that terminology, like why urine is yellow. We should answer this question. All right, so let me write here down first. Why urine is yellow? So I think so you people are known about the spleen, which is the graveyard of RBCs or red blood cells. Alright, so I am going to make here the spleen, a big spleen I am going to make here. This is what? This is a spleen, and here I have RBCs. Alright, because we know that spleen is the graveyard of RBC. So let me write here down as well. Graveyard of RBCs. So this is RBC or red blood cell. Now we know that red blood cell has a pigment which is known as hemoglobin, which is making it as red. All right, it has a pigment which is known as hemoglobin. Hemoglobin. All right, it is broken. Hemoglobin is then broken into two pigments which are known as bilirubin and biliverdin. All right, hemoglobin is breakdown and is converted into bilirubin and biliverdin. So I should write with this color now. Bilirubin, and we do have biliverdin. These are the two pigments by which hemoglobin is made, and when hemoglobin will break down, it will be converted into bilirubin and biliverdin. All right, and these both are also known as bile pigments. These are also known as bile pigments. So these both pigments are also known as bile pigments. So let me make here the liver so that I can make the bile. So this is basically liver in which bile is present, and the pigments of biles are bilirubin and biliverdin. So here it is. All right, bilirubin and biliverdin. So now bile will convert this into urobilin and stercobilin. So we have not need of stercobilin here, but I am still going to write this down. It will convert this into urobilin, and stercobilin. So stercobilin is used in the defecation process. So we are not going to study here, but I am writing here defecation. All right, defecation. Here I am making this as well. This is defecation, and urobilin is a pigment which makes your urine yellow. Urobilin is a pigment which makes your urine yellow, which is used for the urine, and it makes your urine yellow. So here we have gotten the answer why our urine is yellow, just because of the pigment which is known as urobilin. Just because of urobilin, our urine is yellow. So this was all for today. In the next video we will be going with the transport across cell membrane, like how transportation is done across the cell membrane. We would be discussing this in the next video. So this was all for today.